Okay, we're going to get started here.
Great. I'm online.
Great. Welcome. Thank you all for joining us for the inaugural monthly seminar in physical genomics. My name is Benjamin Keene. I'm the Senior Program Administrator at the Center for Physical Genomics and Engineering here at Northwestern. I want to start by letting everyone know that our director, Dr. Backman, unfortunately had to travel today for a family emergency. So I'm going to be moderating the seminar today. Sorry about that. Just as a little background before we get started, the Center for Physical Genomics is a cross-disciplinary research center. Oh, it's Dr. Backman.
Oh.
Hi, Vadim. Why don't I just go ahead with my introduction, though? So anyway, the Center for Physical Genomics and Engineering is a cross-disciplinary research center here at Northwestern that is focused on elucidating the structure and function of the genome through a convergent approach involving super resolution imaging, modeling, computational genomics, biology, and artificial intelligence, with the mission to develop new strategies for the treatment of disease and engineering of living systems, and to train the next generation of multidisciplinary scientists. You can learn more about our research at physicalgenomics.northwestern.edu or follow us on Twitter. So we're very excited to have as our first invited speaker, Dr. Susan Gasser of the Friedrich Miescher Institute for Biomedical Research. When we asked our trainees here at the center to suggest names they'd like to give a talk for this series, Dr. Gasser was the first one to come up. So it's fitting that she's giving our inaugural seminar. Dr. Gasser, at this point, if you would like to share your screen, we can begin your presentation. For our audience, there's going to be time for questions and answers at the conclusion, but you can also type questions into the Zoom chat and we'll get to them after the talk. So Dr. Gasser, thank you so much again.
Thank you. My screen says I can't share until you stop sharing.
Yes, I will stop right now and turn it to you. There we go. Thank you.
Back to the beginning. And whoops. Okay. Sorry. Okay, screen share. Is that fine now?
We can see it, yeah.
Okay, thank you. Okay, it's a great pleasure. I always enjoy, especially if I know that I was selected by the students and postdocs. I'm eager to share our insights. I'm speaking on. I'm not really a biophysicist, kind of an ad hoc biophysicist, but I think questions of DNA dynamics and their physiological relevance is certainly my domain, and I hope you enjoy the talk. So I'm going to talk about how chromatin and nucleosome density alters in response to DNA damage and how that changes what we call chromatin dynamics. I'll explain to you what that means. So I'm going to go back in time to some background. I'd like to tell you about how we initially started viewing yeast chromosomes. And that was 30 years ago with antibodies, and then in the 90s with GFP fusions or incorporating fluorescent nucleotides. So what you're seeing here are old images of staining of a RAP1. It's the yeast TRF1, TRF2 telomere binding protein, and seeing it at the ends of the 16 yeast chromosomes, which are tiny chromosomes compared to any other, even compared to Pombe. And then staining with the same antibody in interphase nuclei and seeing that these 32 dots, actually, because it's a diploid cell, would be clustered into 8 or 9 foci launched our interest in functional organization of chromatin within the nucleus. Also, it's well known that replication foci here incorporating fluorescent DUTP show clustering in S phase. And here you're seeing in white in the middle panel, all centromeres of the 16 chromosomes clustered together at one spindle pole body, and then telomeres from different chromosomes of different lengths at the nuclear periphery, and then in blue here, the nucleolus. So all this led to an understanding that there was actually quite some organization. It's not a random soup of DNA in the nucleus, and that this might be something we could study. So we others also and we as well looked at DNA damaged foci by incorporating arrays of either TET operators or LAC operators and expressing a GFP fusion or a RFP fusion to either a TET repressor or LAC-I repressor, so that we could actually tag DNA sequences and tag them near sites where we could manipulate, either induce double-strand breaks or cause here FRT, a kind of replication block, and then track that in real time in living cells. Now, the yeast is a small cell. A haploid is about 5 microns in diameter. A haploid nucleus is about 1.5 microns in diameter. And these little spots that you're looking at are about 200 microns as the resolution we get. You can actually use deconvolution and get smaller, but these are what we look at. So we wondered how stable these structures are. Just a second, can I get rid of this bar? Well, anyway, I'll leave it at the top. We wondered how stable these different compartments were and how reproducible. And so we started looking over time, and we could see that actually there was a reproducible organization in the nucleus, not just the nucleolus, which is only on one chromosome, but also the positioning of the spindle pole body with respect to new buds, and the fact that telomeres and centromeres had unique positions. We then started tracking individual spots, individually tagged loci, with more and more rapid time-lapse imaging and more resolution of nuclear structure. And then pairs of telomeres ends. So this is right and left telomeres of a short chromosome, single chromosome, and here 2 different chromosome telomeres to see how they behave in space. So the upshot of all these early studies, we were taking 1.5 second intervals. We were seeing a lot of movement. The upshot was to show to us that actually the whole, despite having reproducible, let's say, subcompartments, chromatin and DNA in the nucleus is quite dynamic. So we asked the question, say, let me get rid of this bar. Yeah, can't do it. So we asked the question, how can we track these under a reproducible method in real time? Our favorite, because GFP is very sensitive, is very robust, you can observe it for quite some time, was the LAC-O array with a GFP LAC-I repressor. Sometimes we put up to 256 single binding sites, sometimes many fewer, but it allowed us to observe a locus of interest. And if we monitored this over real time, we could see that we could track this, the movement over, let's say, up to 7 or 8 minutes using 1.5 second intervals. So we wanted to influence its dynamics, of course, and ask what happens when we change the situation. One of the things that we were interested in was DNA repair, because we knew that DNA repair can go on everywhere in the genome, but spatially is often quite localized. So we use this system, and you'll see it again and again, where we induce a single double-strand break. This is a unique cutter, ISC one that'll make a double strand break at a unique site where we insert the recognition sequence, and we do it next to a LAC-I tagged sequence or a TET tag. And then we were taking, at the beginning, 300 3D stacks through the nucleus at 1.5 second intervals with a spinning disk and focal. As time went on, we got better and better at this. And actually, most of the data I'll show you today were done with 750 3D stacks at 80 milliseconds intervals, which cover about 1 minute. Now, the point of doing this, of course, is to try to understand something about the chromatin fiber. So one of my PhD students, Andrew Sieber, was really eager to be able to model this and to go in depth on the chromatin features. And so we teamed up with David Holtzman's lab at the Paris ENS, and Asaf Amitai at that time was a graduate student there. He later moved to MIT. So once again, the influence of collaborating very closely with a polymer modeler was for him to tell us we must really get a maximum number of frames and with the shortest possible time interval. At the same time, we knew that you can do damage to DNA by imaging it. And so we found a good balance between this 80 millisecond intervals. And then what you're seeing here is a single locus. This is not a damaged locus, just a single locus that could potentially be induced for a double strand break. In red, you're looking at individual nuclear pores. You see how quickly they shift around the nuclear periphery. Now, the way we measure movement is to interpolate the center of this nuclear ring. And there's a program actually developed years ago by Daniel Sage, collaborating with us, to interpolate the center of this. And then we measure in each frame the center of the nuclear ring and the position, the radial position of the given locus. We can do it in 3D or we can do it in 2D projections, and then calculate frame by frame the movement. From that kind of measurement, we can extract biophysical parameters. One, of course, depending on the type of movement, but in fact, the confinement or radius of constraint, or in his terminology, length of constraint, is calculated simply by by an MSD analysis of this movement. You can calculate the tethering interactions that hold, let's say, restrain that movement. You can calculate the diffusion coefficient from the slope of the MSD and the anomalous exponent by taking the derivative of the MSD plot. So what this looks like, and the characteristics of the DNA fiber or the movement that we extract from this is either the volume of the space explored, the effect of anchoring, the speed of diffusion, and the character of the movement. Now, I'm sure most of you or all of you are familiar with MSD analysis. This is probably the classic way to study random movement. And the MSD plot is the square of the distance moved in any time interval plotted against the delta t. And a straight line is what you expect for a Brownian random walk to infinity. Now, of course, what we saw within the nucleus was always confined. You had to be confined by the nuclear volume, but it was even confined to much less than nuclear volume. It was confined usually to a radius of constraint of about 0.6 microns, which is less than the radius of the nucleus. And also from the curve, you could estimate whether it was directed superdiffusive movement, truly random movement, or confined subdiffusive movement. By taking the derivative of this, so the log of the MSD over the log of the T, you get a value called alpha. And again, a Brownian walk with diffusion, unconstrained diffusion would be an alpha would be 1. We almost always, well, we did always measure an alpha under 1, and almost never, well, never in the things we were measuring a directed movement. So the conclusion from this was that we were observing confined subdiffusive near random walk in subnuclear volumes. And we could calculate the average. I mean, it would vary from which loci we took, how close they were to centromeres or telomeres, which are tethered. But if we took a normal locus, let's say in a long chromosome arm, we could calculate the length of constraint, the alpha value, always coming under 0.5, the effective spring coefficient, which is the anchoring, we call it, and the apparent diffusion coefficient. So next slide. Yeah. Collaborating with Amitai and Holtzman, they like to model the chromatin fiber as a beta polymer, which you can do when alpha is less than 5. And from alpha, you can calculate a beta value, which is the strength of interaction between discrete particles in the polymer. Now, this is very attractive to someone who studies chromatin because it's very easy to imagine that these are nucleosomes, and that and we know that there can be interactions between nucleosomes that compact and decompact the chromatin fiber. We also knew that we could measure alpha from the measurements that I just showed you, which means we could predict this beta, the interaction between monomers, and we could confirm this by measuring compaction. So everything was in line that we could at least measure, make prediction, and monitor what's going on. So the visualized what we could see, and actually this happened as we induced a double-strand break, was we could see that DNA decompacted. I'll show you that in a minute. And the alpha value, even though it increased, remained under 5. And it appeared that we could model the increase in movement as a decrease in interaction between histone nucleosome monomers. So how could we use this to shed light on a physiological event? Well, the topic I already introduced to you that interested us is how cells repair DNA. And we know that our genomes are bombarded actually with lesions, thousands of lesions per day. We have highly conserved repair mechanisms that repair from yeast to man, and even from bacteria to man in similar ways. But one of the most deleterious is actually the double-strand break. Whereas most other lesions can be repaired without stopping the cell cycle, without interrupting or being coordinated with replication or even transcription, the double-strand break is extremely toxic, because if a replication fork approaches a double-strand break, then it loses. The risk is that the other arm of the chromosome, or the other half of the chromosome, might be lost, and you might be left with a chromosomal fragment and become aneuploid. So now the frequencies of these, as I mentioned, is very frequent, approximately 10 double-strand breaks per cell cycle in a mammalian cell. Now, double-strand break repair can be repaired by end joining, just a lesion, although it's an imprecise repair. And the one that's most conceptually interesting is not just end joining, but homologous recombination. Homologous recombination requires a template from which a resected end, which is processed from a single double-strand break, can invade a homologous sequence, be extended across the break site, and then with resolution of what's called a holiday junction, restore both chromosomes to the intact state. So what happens is that the ends break. They're held together by a complex called MRN or MRX in yeast. The ends, there's a nickase and then an exonuclease that extends, creating a 3' overhang on both sides of the break. And the 3' overhang, once it finds a homologous sequence, will be coated with Rad51, invade, and then promote this extension reaction. This is called the homology search. Now, if cells are in G1, GS, S, or G2, then a chromosome has been duplicated, and the sister is the most common template used for repair of double-strand breaks. However, if both chromosomes are damaged, or you're in G1, or you're in S phase before the sister has been replicated, then you have to find a homologous chromosome or a homologous sequence on another chromosome from which to do the strand invasion and extension. And it's long been argued that homology search is the rate-limiting event in double-strand break repair. So to see this in a visual sense, you have a double-strand break, you have resection coded with RPA, the RPA gets replaced by Rad51, and then if you don't have your sister there to repair from, you go through an exploration, a homology search. That in yeast would mean 1 in 300,000 nucleotides to find a template for repair. Now, in order to do this, the time that this takes, if the sister is not available, is hours. So how does the cell accommodate this? It actually, activates the checkpoint kinase, ATR and ATM, or MEK1 and TEL1, which then block the cell cycle. You don't need both of them actually, but they are usually both activated, and especially after resection. And this gives time for the homology search. And only after repair, then, these kinases are inactivated, the cell cycle resumes, and you can go through mitosis. So what happens at a double-strand break what happens to the mobility of a double-strand break compared to a non-broken site? So we're using a locus on a long arm of chromosome 14, far from its telomere, far from a centromere. We have it tagged about 3 kb away from the double-strand break site, and we track it in real time, either by LAC-I or after the break induction by Rad52 YFP, because Rad52 binds the double-strand break end. This way, we don't have to worry whether we have 100% cleavage. We know if we're tracking a Rad52 site, we're tracking a double-strand break. Now, what we saw was that actually, after the double-strand break, the movement increases. This is shown in an MSD curve here. This is the unbroken site. This is the broken site. The RC here is about 0.46 microns. The RC of this radius of constraint is about 0.7. So there's probably a threefold increase in the volume of the nucleus that each fragment explores, and this in more or less a random walk fashion. So the double-strand breaks are more mobile than undamaged loci, and the increase requires a checkpoint kinase. So we wondered if this was general, and whether only the double-strand break increased movement or other sequences would. So we induced multiple double-strand breaks. First, we showed that by treating them, inducing them by zeaxanthin, which mimics gamma IR, we would get increased movement of loci that were not correlated with a Rad52 spot. In other words, we induced damage, we would see one or several Rad52 foci, but we tracked a locus tagged by GFP LAC-I that was not colocalizing with Rad52 and therefore not a damaged locus. And we could see that the more damage you had in the cell, the more movement there was. And although it never quite reached the movement of the double-strand break itself. So it seemed to be a general effect of increased mobility across the genome in response to damage. It was MEK1 dependent, so ATR dependent, and dependent on a nucleosome remodeler in yeast called INO80. So just to summarize right now, what I just told you is that all DNA moves with a certain small radius of constraint in what is a random or near random walk. It does show sensitivity to metabolic conditions. So in the absence of ATP, chromatin compacts and collapses in a sense. But then when you induce damage in the locus, not only does the locus, damaged locus move more, but undamaged loci tend to move more. So we are wondering, what is it that provokes this increase in movement? And we were wondering if it had to do with an expansion at the break. Obviously, expansion of chromatin, which was predicted from the polymer model, might happen at a double-strand break. So we use structured illumination microscopy. Here we're looking at a phase superimposed on a single focus in G1 or in S phase, and we deconvolved this, use super resolution. So these are not time lapse, these are single points, and asked in multiple nuclei before and after damage if the volume of that single spot increased. And in fact, we saw that it did with increasing damage. This was a TET-RM cherry focus. We could see an increase in volume with increasing DNA damage. We could also do this at a single double-strand break. We could look at the laci next to a double-strand break, induce the cut, measure the volume of that laci adjacent, the laci tag adjacent to the break. And indeed, we could see an increase in volume here, a kind of expansion due to the break. So there seemed to be a change consistent with this polymer model. And another way to ask that, or to ask how the chromatin fiber in general was changing, was to take two tagged foci on the same chromosome arm with no cleavage at either of these sites, and just ask about the relative movement of the two loci relative to each other. Here's just a movie to show you what it looks like. In space, you can interpolate, where the centers of these spots are. They're actually not very far apart. 50 kb is almost our resolution, but you can measure at any given time interval the distance between the two, and then quantify the relative distance between the two over time. Gray here is the uncut situation or undamaged situation. Red is the damaged situation. And once again, we see when we induce general damage in the cell, there's a flexibility induced on the chromosome fiber. Now, is flexibility decompaction? What is it that drives this physical change in the chromatin structure in response to DNA damage? So we felt we had to get down to the mechanism to understand what was going on. Otherwise, it's speculation. So we assume there might be some changes in histones, because obviously that's the major constituent of chromatin. It's known that H2A, and in mammals, the H2AX gets phosphorylated in response to damage. It's checkpoint dependent. Here we see RAD53, the checkpoint kinase downstream of MEK1 and TEL1 getting phosphorylated. So with increasing amounts of zeaxanthin, this is our double-strand break and single-strand break inducing agent. We see an activation of the checkpoint kinase. We see an increase in the phosphorylation of H2A, a sign of damage in every organism, every eukaryotic organism. And then we carefully looked at what happened to H3 and H4. And actually what we expected was some kind of modification. Now, what we saw, and this was repeated hundreds of times, I have to say, because I didn't believe it, was actually a quantitative drop. And this is a Western blot, but it was repeated to be shown that it was in the linear range and repeated many, many times, that there was a drop in core histone levels. It didn't happen to other proteins of the nucleus or cytoplasmic protein. And at maximum levels, after about an hour, it would reduce to about 70%. Now, Western blots are notorious for being nonlinear. So we did SILAC-based proteomics to confirm this. And just in brief, you have two derivatized cells, one with damage, one without, one heavy derivatized, one light. So you always internally normalize. You do mass spec to determine for any single peptide the relative abundance of cleaved and non-cleaved under damage and non-damage. And what we could see was that all 4 core histones lose abundance compared to, this is all other peptides grouped together, and even compared to H2AZ, which is a histone variant that is actually deposited upon DNA damage. So there seemed to be a reproducible drop in histone levels. This was only 20 to 30%, but seemed to be highly reproducible. We then asked, were there specific nucleosomes that were being lost? Was it only at promoters of genes? If you're talking about 20, 30% of the histones being lost, you're still remaining, 80% of the histones. So we could map histones genome-wide. We did this with Owen Hughes, and this is without damage and with damage. And what you're looking at here is the nucleosomal peaks relative to a transcription start site. And the red is the internally normalized to an exogenous seeded in a nucleosome fraction that actually histone abundance goes down all across reading frames, but also in non-coding upstream regions. So in fact, there was no preferential eviction of nucleosomes. It seemed to be a general phenomenon across the whole genome. We asked whether this was just eviction of histones or whether it was degradation. So we used a proteasome mutation, pre-1, pre-2 double mutant, to see if we couldn't degrade histones, if we would ablate this drop in histone abundance. So this is the drop from a normalized, or from the wild type untreated of 1. We see about a 20-30% drop. And if the proteasome was non-functional, we didn't observe that. And we could do the same thing with drugs that inhibit the proteasome. We also asked the question whether it was dependent on checkpoint mutants and on INO80, because the increase in mobility and the expansions was, and indeed we see at least, this is the normal drop in histone level, and we see that it's at least partially attenuated if we don't have INO80, the remodeler, and we don't have the checkpoint mutant. So this drop in histone levels is a response to DNA damage. It doesn't occur only at sites of damage. It seems to occur generally. We see accompanying that an increase in mobility and an increase in flexibility. And just to reinforce the importance of this chromatin nucleosome remodeler, INO80 is a multi-subunit remodeler. It's conserved from yeast to man, has a number of actin subunits, but ARP5, ARP8 contact the nucleosome. And so if you delete either ARP5 or ARP8, the cells are still viable, but the INO80 remodeler is non-functional. And what we see here is that upon damage, this is at a single double-strand break, the wild type increases in mobility, but the INO80 mutant does not. Same thing is true for length of constraint, for the KC spring, the spring coefficient. In fact, it's all consistent with INO80 being necessary for this change. So if I summarize here, what I've shown you is that histone degradation is a regulated process. It occurs after a certain level. It's a fairly high level of DNA damage that provokes the checkpoint kinase. It's dependent on a kind of remodeler that actually we know interacts with the proteasome. It's also dependent on the proteasome, and that it appears to involve histone degradation to a limited extent and chromatin expansion. Now, you could propose that, well, that's very useful. There could be a sense to this because if you have widespread damage, you don't know where it is, or the cell doesn't know where it is, and it wants to maximize its repair. So it wants to increase the accessibility of any potential damage site to the repair machinery. On the other hand, there could be problems with that. You could have lots of random transcription because you're now opening closed promoters by removing histones, et cetera. So there could be a lot of secondary risk in eliminating histones. And actually histones are some of the most regulated proteins in the cell. They're regulated on the level of transcription, on the level of translation, on the level of nuclear import. They're regulated on the level of assembly, and here on the level of disassembly in response to stress. So it's a complex situation. It was really quite unexpected, but we decided we had to explore it yet further. Now, you could also say, well, this is a very unusual situation. You're stressing the cells tremendously and seeing histone depletion, and then seeing these changes in chromatin fiber. Maybe, in fact, this is all an indirect effect of a stress response. So we were very happy to find in the literature that there was a physiological way to reduce histone density. And that comes about by deleting a protein called HMGB1, which in yeast is called NHP6. There are 2 copies of this gene. It's a very important gene. It helps position nucleosomes and maintain nucleosome density. It's a high mobility group protein. Here is its crystal structure in the minor groove, helping in some way to position and load histones. And in both yeast and man, the loss of this protein reduces histone levels, core histone levels. So we asked, this was like nature's gift to us, because this way we wouldn't have to induce damage. We could just ask, in this mutant, which has lower histone levels, almost, well, not quite, but about half as much as we get as a maximal response to DNA damage, what happens to chromosome mobility and expansion and all these things that we measure? And in fact, what we see by tracking intact loci is that they move more in the absence of nucleosome, in the NHP6 mutant, where there's reduced nucleosome density. And we can, using this structured illumination, we can see an expansion of the locus, even without damage. So nature does the same thing in a sense. I mean, has a protein that ensures nucleosome density, but actually you can actually isolate a mutant that is still viable, that has reduced nucleosome density, and it has the same characteristics that we measure after damage. So we could use this mutant and the Inno80 mutant that fails to remove the histones. So the NHP6 constitutively removes histones, and the ARP8 fails to remove histones. And we could ask if it affects homologous recombination. Now, our first assay was simply to look for insertion. It's a big deal if you're doing CRISPR, but it's all in yeast. Yeast is very efficient and doing homologous recombination with linear fragments, even without a CRISPR cut. And so we can measure the rate of integration into, let's say, a mutant Ura locus by integrating an intact Ura locus. And if we do this in an Inno80 mutant, in wild type, you get a certain integration rate. In Inno80, it statistically goes down. Recombination is less efficient. In the NHP6 mutant, where there are fewer histones, it goes up. And if you look for survival on zeaxanthin, this reagent that we're using to induce single and double strand breaks, we see that the Inno80 mutant is very sensitive, maybe because it can't remove histones and repair. The NHP6 mutant is just a little bit more, has a better survival than wild type, maybe repair is faster. But one argument that it does, they are working on the same pathway, is that if you knock out NHP6 in an Inno80 mutant, you improve the survival of the Inno80 mutant. Possibly our interpretation, or one favorable interpretation, would be because there are fewer nucleosomes. Anyway, this was an initial indication. I don't think this is proof, and I'll show you later better proof. Okay, so now I'm going to skip this. This is just a summary up to now. So we wanted to know what's going on with chromatin. I mean, we're seeing fewer histones. Is something else replacing histones? What's going on globally in response to the DNA damage checkpoint and this increased mobility, increased flexibility of the DNA fiber? So we decided to do a whole chromatome or chromatin-linked proteome with and without damage. Now, proteome determination, mass spec, has become much more powerful lately by using this TMT labeling that allows you to post. You no longer have to actually label the cells like in the SILAC. You can post isolation, label the peptides, each one with a different tag with a range of tags, and do quantitative analysis of thousands of peptides, whereas before it was always much more limiting in what you could detect. So what we did was take yeast cells with and without zeaxanthin. They were blocked in G1, so we're only looking at one phase of the cell cycle. We lysed them, cross-linked them, and did chromatin fractionation through sucrose to get rid of soluble proteins. So we're only looking at chromatin-bound proteins. It's not 100%, obviously, because we don't really want to get rid of too many proteins, but it does give a separation from cytosolic proteins, and ribosomes and so forth, which are much more abundant in yeast, and allow us to have a much more sensitive view of chromatin-bound proteins. You then digest those to small peptides, TMT label them, and do quantitative mass spec. We did this with and without damage in wild type yeast, in the INO80 deficient yeast, because there we expect not to see the change, and in the NHP6, which may already have some of the change that may increase mobility. So now I want to just use this slide to impress upon you how powerful it is to do subfractionation of the chromatin prior to doing mass spec. This is the total proteome analyzed in exactly the same way, with and without damage, wild type ARP8, and looking at the fold change difference of either increase or depletion. And there are very few proteins that actually significantly increase or decrease in the total proteome. Now, that's not because they don't change, it's that it's kind of swamped out by the vast majority of cytosolic proteins that don't change. Because when you look at the chromatin changes, and this is, of course, many fewer proteins, but now you see about 750 proteins increasing their abundance and about 560 proteins going down. And the same in ARP8, somewhat fewer in NHP6. So this gave us a sensitive way to look at what's happening to all chromatin proteins, or the abundance of chromatin proteins in response to damage. Reassuringly, we saw histones, core histones, all go down in wild type. This is in wild types in red, and not in the INO80 mutant. That's the same thing we had seen for all of our phenomenon. And in the NHP6, it's a very attenuated picture, and that may also be what we expect since they're already depleted. So your 0 point here is already lower. We also saw that DNA damage triggers loss of all 3 RNA polymerases, POL1, POL2, and POL3, and these are shared subunits. Now, this is really interesting. It means you're downregulating transcription. This was known that transcription is downregulated in response to damage. But here what we're seeing is actually an eviction or loss of the polymerases from the chromatin, all except one subunit, which is actually involved in repair. So this may explain why we don't see massive transcription or promiscuous transcription following damage. When we looked at the proteasome, I showed you already that this degradation was proteasome dependent. we see the subunits. Almost every subunit goes up, reassuringly. The proteasome is nuclear and does associate with chromatin. And when we looked at ubiquitin ligases, which would be necessary for ubiquitinating proteins for the proteasome, we also saw, well, a mixed bag. Some E3 ligases went up, some went down. There's actually a whole zoo of these ubiquitin ligases. And actually, it's not very clear to us, or was not clear, which ones might be involved in repair. So we took a global view of this. We mapped the behavior of all the E3 ligases, E1, E2s, and this large family of E3 ligases. And what you're seeing here is a heat plot. The red is enriched on chromatin. The blue is depleted from chromatin. So you see E3 ligases going in each direction. And the second column is N080 mutant. So what we were particularly interested in would be any E3 ligase that would be up on damage in wild type and less up or not up at all. This is a good example in the N080 mutant. So we were looking for N080 dependent ubiquitin ligase changes. And we noticed that there were a number that fit that characteristic. Now, interestingly, they did not overlap exactly with the set of ubiquitin ligases that were already known to be involved in histone homeostasis. I told you that there is tight control of histone levels in the cytoplasm, and a number of E3 ligases had already been involved in excess histone degradation or implicated in excess histone degradation under different conditions, but in the cytoplasm. Now, we were looking at nuclear depletion because this is chromatin associated and it happens within a half hour of damage. So we're not looking at newly synthesized histones. And so we could characterize some of these and we'll compare them what happened, but many of them did not accumulate on chromatin, the ones marked in blue here. Some of them did not show N080 sensitivity, the ones marked in orange, and some of them did. But only 3 of those identified were actually known that they might affect histones, and there were a lot of unexpected hits. So we were left with the only option is to test each one of them one by one to see if they respond to zeaxanthin to degrade histones. So this is a summary of a lot of work of something called HiBit, where you can measure the level very quantitatively from cell extracts by derivatizing the proteins after they're on the blot. And this is done with histones. And what you see here at 3 time points, at 0 time point, 1 hour, and 2 hours of zeaxanthin, you see that the level in wild type cells of H2B, but we did it on 3 different histones, goes down. And in the ARP8 mutant, it doesn't. It stays, I mean, it goes down slightly, but it's attenuated. And so we tested these one by one knockouts of actually about 25 E3 ligases. I only show you a few here. But the 5 that we saw that were N080 dependent enriched on chromatin are these boxed here, and all of them attenuated the depletion, particularly at 1 hour of histones. And others that were implicated in the cytoplasmic turnover of histones were not. They looked just like wild type for the degradation of chromatin-bound histones. So we have a good indication of at least 5 E3 ligases implicated in histone degradation. Now, what are these 5? Here they are. And what is their target? Well, only one was really known what it targeted. That was the BRE1-RAD6. It's an E2-E3 complex. These work together. It's the RNF2040 homolog. It targets K20 on H2B C-terminus and is a monoubiquitin ligase. The monoubiquitin is used as a recognition site for binding proteins, but not for degradation. The others were known, RSP5, UFD4, PEP5, to be polyubiquitinating enzymes, but it wasn't, they weren't implicated in anything to do with damage. So we asked whether they are actually recruited to double-strand breaks. So we tagged them, we induced a double-strand break and did chromatin IP to ask if they're actually binding at a specific double-strand break. This is the increase, and all of these 5 ubiquitin ligases are recruited. The only one that doesn't show enrichment is UBA1. It's not one of our E3 ligases, but it's the E1 enzyme, and it is not bound at damage. So their recruitment's enhanced by additional damage to this double-strand break, and it depends on the DNA damage checkpoint and NO80. So we have a redundancy of E3 ligases that seem all to affect in different ways histones, although the end product is that they lead to a partial depletion of histones, generally in response to the DNA damage checkpoint. Now, the question then is, why so much redundancy? Well, I already hinted at it. We know that BRI1 Rad6 will do monoubiquitination, but not poly, and monoubiquitination will not lead to degradation. So PEP5 and RSP5 are known to polyubiquitinate, not monoubiquitinate, so they may be acting on the same lysines. And UFD4 is both a mono and poly, and it may be acting yet on other known lysine targets. However, we don't know, and we weren't able to map which are the targets of these novel damage response kind ubiquitin ligases. So the next question was, does it matter? Now, you remember I told you what was rate limiting, or what we thought was rate limiting, was homology search. So our real question is, does loss of histones and loss of the, or does loss of histones facilitate strand invasion? And if you get rid of the ubiquitin ligases that block the loss of histones, do you attenuate strand invasion? In other words, does the expansion and flexibility of chromatin fiber increase DNA repair? So I'll cover this in just 3 minutes. So the answer is yes, but I'll take you through the assay. We induce a double-strand break on one strand of the chromosome of on one chromosome arm of a partial gene, a lys2 gene that is only the N-terminus of the gene. And the rest of the gene, the C-terminus, the lys2 gene, is on an unrelated chromosome. So that repair of this double-strand break has to occur by a homology search and recombination with this other arm of chromosome 11. The cut is on chromosome 5. We can monitor this strand evasion event by PCR, by using a primer that is specific for one chromosome and the other chromosome. And if you have not done strand invasion and elongation, you will not be able to get a P1, P2 fragment, which is here in the red arrow. Now, I told you the homology search takes a couple hours in wild type, and indeed what we see here after the HO cut induction, it takes 3 to 4 hours to see the first sign of this P1, P2 fragment. In other words, completed strand invasion. This is a control fragment down below. If you get rid of pol32 or ARP8, you see a drastic reduction. Pol32 is a subunit of pol delta. It's known to be involved in this strand invasion. And if you get rid of NHP6, which gets rid of histones, we actually see an advance, an earlier, slightly earlier strand invasion. So this is quantified here. NHP6 mutants are a little bit better than wild type. ARP8, pol32 worse. So now what about the ubiquitin ligases? So we did the same assay in a PEP5, UFD4, and Rad6 mutant. BRI1 we also did. And it's clearest here in Rad6. You see that there's really, this is the monoubiquitinase. You see a really drastic drop in strand invasion. It's quantified here. The Rad6 and pol32 are almost the same, very poor strand invasion. The others show a partial effect, which is exactly what we expect because they show a partial effect on histone degradation. Now, interesting, H1, the linker histone H1, is also a target of ubiquitination and degradation. I haven't shown that to you, but it is. And yeast cells are viable without this linker histone because yeast chromosomes don't condense very much. And if we get rid of this linker histone, just like when we get rid of NHP6, we see a slight increase, especially at early time points of strand invasion. So in fact, you can shift the rate, or let's say the rate of strand invasion with the density of histones. Not only that, we could ask what happens if we combine a ubiquitin ligase mutant with the NHP6 so that you're counteracting the inability to remove histones with having lower element levels. So NHP6 is here in green, RAD6 is down here. If we combine them, we see that we, improve the inability to do strand invasion by getting rid of histones. So there's still one question left, but I'm not going to go through the data. Is it template accessibility or is it really movement that makes the difference? And we have one bit of evidence that it's actually template accessibility and not break movement because we have a mutant that actually allows the break to move, but doesn't affect, or no, it's the opposite, allows the external nucleosomes to be removed, the ectopic, but not the local, movement. And we still see wild type recombination, suggesting that it's actually the template accessibility and not necessarily the movement, and that movement is a side effect. So I won't show you that because it's a rather detailed study, but you can find it in our paper, our 220 paper actually. So in summary, I've shown you that DNA damage triggers a high level of DNA damage, triggers about 20 to 30% drop in histones and H1. It's checkpoint kinase, NO80, and ubiquitin ligase dependent. Histone loss increases DNA flexibility, fiber flexibility, and histone loss speeds up strand invasion. So it also can be used artificially. You were talking about engineering to increase CRISPR-mediated HR. We've tried it, but it wasn't as successful in mammalian cells. So it's something that requires work. But there is a displacement of histones as well in response to damage, but there's much less degradation. And I'll leave you with this beautiful image, this model of what happens when you remove one nucleosome to the length of the DNA fiber. You see it increases 7-fold, but it's really easy to imagine that the loss of one nucleosome can increase the flexibility of a chromatin fiber if the loss of one nucleosome does that. So I'm happy to take questions and hope you enjoyed the talk. I'd like to thank my lab, which no longer exists. So I'm now retired and I closed my lab, but I'm working in translational cancer research to promote the impact of this kind of basic research on cancer treatments, including radiation biology. So thank you very much for your attention, and I'm happy to take questions.
Thank you so much, Dr. Gasser. That was incredible. We're coming up on the hour, so if anyone would have a question, why don't we, here, we've got one in chat right now.
Do you know whether histone loss is by the entire nucleosome? That's a good question. We know that we usually follow H2A and H2B, but we have followed by fluorescence H3 and H4. And it seems that they all behave quite similarly in bulk. Whether that means that the whole nucleosome is evicted, we don't know. It's a good question. Does euchromatin and heterochromatin respond the same way? That's also a good question. Budding yeast, the only real heterochromatin, and some would debate that it's real heterochromatin, is Sur-coated nucleosomes. at telomeres. We still see some loss at telomeres, but it's interesting. Access to heterochromatin is dependent on the SWI/SNF nucleosome remodeler, much more so than in 080. So I'm thinking that maybe there may be a slightly different mechanism, but I think there is still an ability, there's certainly ability to repair double-strand breaks in heterochromatin. Now, in mammalian cells, I guess you're aware, the double-strand breaks in heterochromatin and in flies are relocated out of heterochromatin prior to resection. So in a sense, there's already, let's say, a change in heterochromatin accompanying damage that will make it able to be more accessible and probably accessible to this kind of histone attack. You're welcome.